other student's submission. Ideally, somebody that doesn't have a peer review associated with theirs already. Just leave a comment, you know, hey, good job. Maybe do this to improve, or hey, this looks really good, it's really understandable. The primary question you should be asking is like, does it thoroughly explain the concept? Okay. So just take a look at that. And then based on peer review, make edits to yours as necessary and submit your final draft by Sunday. For lab, you'll notice something was loaded in yesterday, so the simulation was not fully complete, but you do have a labster this week on cellular respiration. So this says it's not yet posted, but it is posted now. You can see it in the modules. I can show you, if I pull up our modules, if you scroll to week 8 and you scroll down to lab, you'll notice it should be in there. Where'd it go? Okay, well, the labster is not posted. It should be posted. Let me look at this. Did it maybe just not go out of here? Oh, here it is. My goodness, sorry. So you do have a labster. This is due by Sunday. So I know that a lot of you like to get ahead on work, which is phenomenal. I encourage that, you know, time management. The big thing with this labster is that I really would recommend getting through the lectures and getting your concept checks for the lectures out of the way first, because the labster covers cellular respiration, and unless you've kind of watched the lectures and feel somewhat familiar with cellular respiration, the labster will be a big challenge. So again, what I would really recommend doing, if possible, is saving that labster until after you have watched the lectures, done the concept checks. I think that in your recommended schedule on our homepage, I think I might have popped it in there. I popped it in there on Friday. Again, that's just a recommendation, but the reason I recommend it for Friday is just because ideally at that time you will have finished the lectures and you will have finished the concept checks. So have a decent understanding of cellular respiration. The reason we're putting a lot of supplemental stuff in with cellular respiration is very similarly to how cell communication was. Cellular respiration tends to be one of the sort of tough spots in Bio 181. And we just want to make sure that you have enough supplemental information to truly master this concept. Mastering cellular respiration, mastering cell communication will really set you up for success for classes after Bio 181. So although all learning objectives are important in this class, there are certain ones that will really be good foundation material for moving forward through your majors. Cell respiration, cell communication, those are two major ones. So we just wanted to make sure you had a lot of support. I know some of you think that the labsters are creepy, and I agree. They're all we have available for our for like decent lab, like online lab simulations, but I can't agree more. Like they're creepy. But it's October. It's like creepy time anyway. All right, so individually labsters, this is posted officially. Make sure that is an individual, you have completed the lab video enzymes as well as the pre-lab quiz. So this originally had a due date of yesterday, but I think I ended up moving it to today so that you had just a little bit more time by 11:59 p.m. And then when you meet with your lab professors this week at your scheduled lab time, you'll be working toward so we're done with osmosis, and so you'll be working toward a new lab, a new series of labs focused on writing and planning, and that has to do with enzymes, which again are a very, very important learning objective from Bio 181. So we'll learn about enzyme kinetics, and you will conduct parts 1 and 2. So your lab instructor will give you more directives on that. So that's just what to expect this week. Fun stuff. But what I want to do is move forward to, whoa, where I zoomed past, which is our muddiest points. Before I get there though, I do want to make sure to ask, are there any questions about just like what's going on in lecture or lab this week or anything else? If you have a question, unmute yourself, raise your hand, anything. Otherwise, we'll charge forward.
I have more so like a comment.
Yes.
Thank you for letting this week be a little bit lighter in bio compared to other weeks because midterms are stressing me out.
No, I know we kind of thought about that, and also just after what happened with the, which again, you guys did amazing on the Unit 1 checkpoint, but I also know that that caused a lot of stress. So I never want to do that. So we were kind of like looking at this, and we were like, let's take it easy. Take it easy. So yes, but thank you. I'm glad it's helping. Yeah. Any other questions or anything? All right, well then let's jump into the material. I have to say, you guys had amazing questions. I'm pumped to talk about cell signaling. I love cell signaling. Okay, but I hated it when I was in undergrad. So if you're like, I hate cell signaling, I understand. Let's make you love it. All right. Okay, so first off, cell signaling is a lot of times when you hear the term cell signaling or signal transduction or cell communication, it's referring to the same thing, which is always a joy when there's multiple terms that mean the same thing. But basically, whenever you're talking about signal transduction or cell communication or cell signaling is really how you'll probably hear about it mostly. It occurs in 3 distinct steps. The first thing is that cells communicate primarily through receptors and ligands. So the ligand or the signaling molecule is basically going to be the message that needs to be communicated. So that's your ligand or signal. And then how a cell like hears that message is through receptors. So as we will talk about later today, there are a variety of receptors, but basically what happens is when the cell hears that message, it can basically like take that message and interpret it in a variety of ways. So sort of like your body, right? You can interpret message that is that you hear, you're just listening and processing and reacting, right? Kind of like, okay, cool. Or you can maybe like a similar like reaction is the message you're receiving is like heat. For if you example, you put your hand on a hot stove, your muscles are activated to like pull back immediately. So those are different responses like listening and hearing versus, you know, like the reaction to burning yourself on heat. So that's sort of like receptors have different functions based on the message that they're hearing. So we'll talk about those functions a little bit later as well. So the message is the ligand or signaling molecule. The receptor is what hears that message and kind of starts the like process of interpreting it, that message. Usually what happens once that message is heard is you will have a series of reactions. Again, sort of like if you put your hand on a hot stove, you feel pain, but also you have the reaction where your muscles contract to pull your hand away. So that's multiple messages, and that's interpreted with the nervous system, the muscular system, etc. And so similarly, when a message is heard by a cell, heard by a receptor, there's a series of reactions that take place, a cascade of reactions that take place that lead to the response. So there's a variety of reasons that we have a pathway of responses, like why isn't it just the receptor leads to a response? Why does it have to be so complicated? And the answer is Well, one, if you have multiple steps that lead from the receptor to a response, you have a lot of time between activation and a response that you could intervene. So if all of a sudden you're initiating a response, but you want to stop that response, like let's say the stimulus stops, you can stop the process right here or right here, and then you won't ever get to the signal or the response. So multiple steps means multiple levels of regulation, or basically points of intervention. Another reason is sort of what was mentioned, I think primarily in the lecture, is that what can happen with a signal transduction pathway, especially like the protein kinase pathway, which we'll talk a little bit about, is that you can get what's called signal amplification. So even though you may have one signal or one ligand bind this receptor, a signal transduction pathway would enable like maybe this molecule to cause a change on this, like 3 of these purple ones. And then these purple ones will activate like, 7 of these green ones, right? So that's what's called signal amplification. Even though you may have one receptor, that can lead to the production of lots and lots and lots and lots and lots of second messengers, which would lead to that signal even just be coming from like one ligand. That signal would be very, very loud, sort of like a loudspeaker. So that's another purpose of the signal transduction pathway. So multiple steps for intervention as well as signal amplification. So number 1 in cell communication or signal transduction or cell signaling is that you have a ligand or a signal heard by a receptor. And then that message is communicated through the cell via signal transduction. So that's just a pathway of communication from receptor to response. And then, of course, number 3 is the response. There can be lots of different responses. These summarize like primarily 3 of them. You can have a chemical reaction be a response, like an enzyme, like, hey, here's lactose. We need to make lactase to break it down. Or it could be structural proteins, like, oh, hey, here's a bacteria. We need to create pseudopodia and do phagocytosis and eat it. Or it could be a transcription factor, and transcription factors activate gene expression. So they turn genes on or off, which we'll talk more about in about 2 weeks. So don't worry too much about transcription factors yet, but just know that that's kind of interacting with DNA to turn genes on or off. But yeah, so lots of different reactions. So you just have the message, which is heard by the receptor, communication of that message to the response. So that are those 3 steps. So what I want to talk about first, which seemed to be one of the most common points of confusion, which is how do cells communicate with each other? So how does one cell send another cell this message, this ligand? So there's a couple of ways that this can be done. You can have what's called direct intercellular signaling. Anytime you hear the term inter, you should think inside. So we'll talk about that. And then you can have what's called contact-dependent signaling. You can have autocrine signaling, paracrine signaling, and endocrine signaling. I want to review these. Okay, first, again, whenever you hear the term inter, so this is direct intercellular signaling, you need to think inside, right? So with intercellular signaling, this is direct cell-to-cell communication, but more specifically, this is when the message is passed directly from cell to cell, basically from cytoplasm to cytoplasm. So in this case, this is sending a message from cell to cell through gap junctions. So if you remember when we talked about cell junctions, we talked about gap junctions in animal cells and plasmodesmata in plant cells. These are essentially the same thing, which is if you have 2 cells, you can see like these 2 cells right here lined up next to each other. Gap junctions in animal cells and plasmodesmata in plant cells are essentially just little like tunnels that lead from cell to cell. And so what that means is if this cell right here is trying to communicate a message, it doesn't actually have to like secrete that message out of the cell. If that message is needing to go to cells within the same tissue, like right here, this cell can communicate that message directly from its cytoplasm to the cytoplasm of the adjacent cell through these tunnels, through these gap junctions. So with direct intercellular signaling, that message, that ligand, is passed directly from a cell through a tunnel, through a gap junction or a plasmodesmata, into the cell next to it in a tissue. So here, what you're looking at is kind of what we always talk about when we talk about with gap junctions. This would be, oh, well, actually, I think this is ciliated columnar cells. So I think this would be cells that line the trachea. So this is a little bit different than what I was thinking. I thought it would be like stomach cells. Well, let's pretend these are cells in your intestine because we've talked about those. So when we talked our cells, sorry, we talked about this in cells in your muscle. A lot of cells in tissue have gap junctions. But we talked about the fact that like muscle cells have to get stimulation from one nerve. But like, for example, in your muscles, like in your bicep, you have tons and tons of muscle cells. So even though you may have one neuron stimulating your bicep, all of the muscle cells in that bicep have to get the same message in order for you to be able to, flex. And so what happens is, let's say we have a neuron stimulating this cell right here in the middle. That neuron says contract. We don't only want this cell to contract, we want all of these cells to contract. And so in order to get that message, this cell creates a response or a ligand, and that is sent directly through gap junctions to all of the cells in this tissue. So even though this cell is saying contract, it's sending messages to all the other cells in this tissue that they should also contract. So direct intercellular signaling is when you're passing a message cell to cell through a gap junction, through a tunnel. So they're sharing essentially cytoplasm. So that message doesn't even have to leave the cell. Okay, so that's direct intercellular signaling. We also have what's called contact-dependent signaling. And this is different because in this case, it is spreading a message cell to cell. These cells do have to come into contact with each other, but the message is not spread through like a gap junction or a plasmodesmata. Instead, you have this cell right here with its message. So the little message is in blue. And then you have this cell, the target cell, with the receptor, which needs to hear that message. In order for this cell to hear that message, this cell has to come up to the cell and touch it. Okay, so I like to think of contact-dependent signaling as sort of like a handshake, right? These cells have to like shake hands. They are communicating to each other like who they are, like here I am, hi. They're touching. Handshake requires touch. And that is what communicates that message. This is direct cell-to-cell communication, so similar to intercellular signaling, it's cell-to-cell direct via contact communication, but intercellular signaling happens through gap junctions, whereas contact-dependent signaling, these are plasma membrane proteins. These are integral proteins. So you have a protein expressed by this cell, you have a protein, a receptor expressed by the target cell, and these proteins touch each other. And what would happen is when that protein touches this receptor, this cell, communicates in response. Again, these cells are not connected to each other, but they do have to touch each other for this to happen. Kind of like a handshake. So I want to mention something, actually give you a really good example of when contact-dependent signaling is really, really important. And Contact-dependent signaling is extremely important for tissue growth, tissue regeneration, and actually defects in contact-dependent signaling are one of the contributing factors to tumor formation and cancer. So let's talk about it. So first off, kind of just to like show you what you're looking at here. This would be an epithelial tissue. So you have cells lined up next to each other. Just like any other tissue, epithelial tissue, that's sort of what you see, right? Cells lined up next to each other. And this is showing the same thing here over to the left, but this is specifically just kind of like simplifying it off to the right. But off to the left, this is looking at like sort of like what skin looks like, right? So you have multiple layers of epithelial tissues. So let's say you get a wound. So this is quite the deep wound, but let's say that like what should be happening normally is this skin would be like completely across, right? So it would be homogeneous here. But what happened is this wound led to like a big chunk of the epithelial cells, you know, missing here. That's essentially zooming in on a big cut. Okay. So one thing that has to happen, right, if you have your skin and you get a big cut, is you have to take that cut and you have to make the skin come back together. So those skin cells on either side of the wound, here and here, have to grow together until that wound is sealed up. Okay. So how cells do that? I guess like what I should say here is that those cells have to grow together to seal the wound, but importantly, they have to like stop, right? Because what would happen is if we have these cells here, these cells growing inward this way, these cells growing inward this way, is eventually you would have cells grow to fill in that wound and then stop. But if they didn't stop, and if they kept growing, you would get cells on top of 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 cells, and then you would get a mass on the skin. And that but that is what you call a tumor, is when you have cells grow and grow and grow and grow and not stop. So again, that can happen with skin. So for example, if you get a wound, deep cut, cells are growing in this way, cells are growing in this way, they should stop when they contact each other. But if they don't, and they grow and grow and grow and grow until where you have like a mass of cells, that's technically a tumor. This is what we call contact inhibition. Basically, when cells will grow and grow and grow and grow until they touch each other, and then they stop. And you can see that over here on the right. You again have a tissue, let's say epithelial tissue, and let's say that this was a cut or something like that. This is just like how a normal tissue, or like an epithelial tissue, or like a vascular tumor or something like that, would form, is one of these cells all of a sudden loses that contact inhibition. So basically the fact that when they're next to each other, they shouldn't grow anymore. And all of a sudden that cell starts to grow out of control. And that is just one. There are several contributing factors to tumor formation and cancer growth. But this is a really big one, is your cells should only grow in a tissue until they kind of like have the correct structure. Remember, structure equals function. And so if a cell grows out of control or keeps growing even though the tissue has its normal shape, this mass of cells starts to impact structure, which starts to impact function. So again, this is an example of contact-dependent signaling. Specifically, this is called contact inhibition, when cells will grow in a tissue until they contact each other, and then they will stop. All right, I hope that makes sense. Are there any questions about this? If you have questions, feel free to raise your hand or unmute yourself. Okay. If not, we will move forward. So that's contact-dependent signaling. Again, intracellular signaling is passing the signal through the cytoplasm. Contact-dependent signaling is just these cells contacting each other, but it does require like not passing the signal through gap junctions and instead just having a ligand bind to a receptor. Again, kind of like a handshake. Now, it looked like the biggest points of confusion with these terms were between autocrine and paracrine. So here is where the big difference is. And what you need to like remember is that anytime you hear the term auto, for example, like autocrine or autobiography, you're thinking of like with an autobiography, that's when somebody writes a book about themselves, about their lives. And with autocrine signaling, this is cell communication where the cell is communicating with itself. So this may seem weird, like why would a cell do this? But one example of this would be like, we talked about like plant infections and the necrotic spots. If a cell gets infected with a virus, it wants to kill itself, basically. The cell needs to undergo programmed cell death. And what happens is that cell basically says, I have a viral infection. I need to form a necrotic spot to stop the spread of this infection. So autocrine signaling is exactly that, right? So one cell creates a message and hears that message. It's talking to itself. So that's what we're looking at in this picture, right? So you have the cell creating the message. The message is in blue. Notice that that cell has the receptor to hear that message. That means that this cell is creating that message and it's hearing that message, right? It's talking to itself. However, autocrine signaling also can happen, like you'll notice where this cell is producing a message and not only is it hearing the message itself, but all like nearby cells are hearing that message as well. But these cells are in close proximity. Another thing to note is that these cells are the same, right? They're all yellow with purple nuclei. So this cell is communicating with itself, but it's also communicating with nearby cells of the same type. But again, how do you recognize autocrine signaling? It's when a cell is communicating and one of the cells it's communicating with is itself. But it can also be communicating concurrently with nearby cells. But importantly, one of the cells receiving that message is the cell itself. I mention this because if we compare this to paracrine signaling is when a cell is producing a message to nearby cells, so very similar to autocrine signaling, but notably, this cell is not hearing that message, right? So it's nearby cells, but the cell is not talking to itself. Okay, so again, autocrine signaling, this cell is producing a message and it's hearing that message. It's talking to itself. It also may communicate that message to nearby cells. Paracrine signaling, this cell is communicating a message to nearby cells, but that cell is not one of the cells hearing that message. So I say like autocrine signaling is like talking to yourself. Paracrine signaling is talking to others who are like in the same room, right? So if somebody was sitting next to me and I was talking to them. Autocrine signaling is like, man, I really need to drink more water. Paracrine signaling is, you need to drink more water. Okay. Now, I do want to mention, I thought I was moving to endocrine, but let's take a break. This was a point of confusion, was one of the concept checks asked about synaptic signals. Synaptic signals, which are signals that are sent from neurons, and these signals can be received by other neurons or like muscle cells, or actually any tissue, right? So neurons stimulate themselves and everything else. But importantly, neurons don't actually come in contact with the tissue that they are sending the message to. So if you look here, you have the signaling cell, so the neuron. It's releasing a message. These are neurotransmitters released via exocytosis. But notice that these are actually released into a space. So no matter what, neurons have a space between the neuron sending the message and the tissue or cell receiving that message. And that here is just referred to as synapse. Sometimes this is referred to as the synaptic cleft. Regardless, it is a space between the neuron and whatever's receiving that message. A small space. These cells are close by. And so when this message is received by a close by cell, that's paracrine signaling. Again, that's characteristic of one cell communicating a message to close by cells. All right. Okay. So synaptic signaling, neuronal signaling is paracrine signaling. Now, endocrine signaling. So remember we said autocrine signaling is like, ooh, I need to drink more water. Paracrine signaling is like, you need to drink more water. Endocrine signaling is when one cell is communicating a message to another cell over a distance. So you see here you have this cell creating a message, and that message is being spread through the bloodstream, or maybe like the lymphatic system, or some sort of like distance mechanism to a target cell. So again, this is like messages being conveyed over distances. So this, I would say, is like a phone call, as opposed to me saying like, oh man, I need to drink more water, which is autocrine. Paracrine would be like me talking to somebody in the same room, you need to drink more water. Endocrine signaling would be like me picking up the phone and calling you wherever you are, Arizona or somewhere else, right? I'm in Missouri. Presumably none of you are in Missouri with me, but picking up the phone, calling you, and telling you to drink more water. So those are the different types of signaling. Again, I would distinguish them: direct intercellular signaling, that is cytoplasm to cytoplasm via connected cells. Contact dependent signaling is cell to cell, like a handshake. Autocrine signaling is the cell talking to itself. Paracrine signaling is the cell talking to a nearby cell, but over a space, a small space. And endocrine signaling is cells communicating over large distances. But again, all of them are doing the same thing, which is conveying a message. Okay, so now they have that message. Now what do they do with it? So that's where we move on to how these messages are received. And messages are received via cellular receptors. But before I go into cellular receptors, I want to make sure that everybody is feeling okay about cell-to-cell communication, like the difference between autocrine, paracrine, la 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 la. How's everybody feeling about it? Okay. Are there any questions so far? Again, like always, feel free to ask questions, not only out of like confusion, just curiosity is also encouraged. But if you feel good, we can totally move to receptors. All right, I don't see any questions. So let's move to receptors. Feel free to ask questions as we go. All right. Okay, so again, these messages are interpreted by receptors. We have two kind of like primary different types of receptors. The first of those would be a cell surface receptor. So this is just like an integral protein that's in the plasma membrane. Or you can have receptors that are actually inside of the cell. These would be intracellular receptors. So either punched in the membrane or floating around in the cytosol. Cell surface receptors are going to be like the most prevalent These are integral proteins, meaning that they pass between the outside and the inside of the cell because the message has to be received on the outside of the cell, interpreted, and that message has to be sent inside of the cell. And so these are going to include enzyme linked receptors, G protein coupled receptors, ligand gated ion channels. And there's some other ones that are a little bit more rare, but those are your three primary types. And then you have intracellular receptors, which are pretty much one type. So let's go through each of the cell surface receptors. So first, we have enzyme-linked receptors. And a really good example, and I think the primary example that your book gave, were protein kinases. So importantly, a kinase, anytime you have the suffix ase or ace, you need to infer that that is an enzyme. So this tells you it's an enzyme. Importantly, kinases, what they do is they add a phosphate group to something. And anytime you add a phosphate group to something, that's called phosphorylating something. Okay, so if you have watched the lectures from this week, yay, good job. But this week, we are talking about cellular respiration. And cellular respiration has a really important goal of creating a ton of adenosine triphosphate, or ATP. So you can actually look at this picture over here, kind of going in reverse of what we're talking about. But when it comes to ATP and cellular energy, ATP is sort of like a rechargeable battery, and you charge it with phosphates. So here you have adenosine diphosphate, which means it's adenosine, a little ribose group, and you can see these two little orange circles here. Those are phosphate groups. So you should think of ADP, or adenosine diphosphate, as an uncharged battery. You can charge the battery by adding a third phosphate to it and making it adenosine triphosphate, or ATP. So again, this week you're talking about cellular respiration and a really long process that will essentially just give you like a ton and ton and ton of ATP, like fueling phosphorylation. But that pathway is not the only way that we can get phosphorylation. You have enzymes like kinases that can actually add a phosphate to a molecule. So for example, here, this usually does require ATP, which is where we actually like steal the phosphate from. So for example, here in this picture on the right, you have an inactivated receptor, so a receptor that doesn't have the ligand bound. And then inside of the cell, you have just like some protein or some molecule. Okay, so don't even go into depth about what it is. It's just like something. But what you need to know here is that lots of enzymes and lots of proteins are essentially like turned on, like their function is turned on by having a phosphate group added to them. So how you can kind of think about that is that whatever this protein is, if it's unphosphorylated, functionally, it's off. So if we want to use that enzyme, we need to turn it on. So what can happen is you have this receptor. It's an enzyme-linked receptor, meaning that the intracellular portion of that receptor has an enzymatic function, which, remember, enzymes just perform chemical reactions. And in this case, since we're using the example of a protein kinase, it turns out that the enzymatic function of this receptor is to add a phosphate group to something, to phosphorylate something. So we have this receptor. Once we have our ligand or signal bound to the receptor, that basically activates that enzyme. So you'll notice here it's just like green, but when it's activated, it's pink. It's like glowing. It's on. It's an enzyme. Okay. And again, that enzyme phosphorylates. And so you can see here, we have a ligand bind that receptor. It turns that receptor into an enzyme. and in particular, protein kinases. Kinases phosphorylate things. This removes a phosphate group from ATP and adds that phosphate group to this protein. That creates ADP because we took off a phosphate group. So whenever we remove a phosphate group from ATP, that creates ADP. When we use ATP in a reaction, that is using energy. So this is an energetic or energy requiring process. But regardless, what happens is this enzyme is directly, it's an enzyme linked receptor. This receptor is linked to an enzyme. And in particular for protein kinases, what these enzymes do is they phosphorylate something. So that's just what you're seeing here. And again, why does that matter? Phosphorylation is an extremely important mechanism of essentially like activating or turning on things in a cell, especially enzymes. So protein kinases are going to be one of the most important like enzyme linked receptors that a cell has, because activating proteins is a really important function. So enzyme linked receptors are receptors. So I want to break this down a little bit more. If we look at this picture, technically the receptor is this little like U-shaped molecule right here, right? Because the receptor is binding the ligand. And this is the enzyme. That's why we say it's an enzyme linked receptor, because it's a receptor linked to an enzyme. And again, it's the enzyme that actually has the catalytic function, or it's the enzyme that basically drives the chemical reaction. And in the case of our example, which is protein kinases, the reaction we're driving is phosphorylation. All right. So our process for any enzyme linked receptor is just we have our ligand find a receptor. That receptor has enzymatic function or is linked to an enzyme itself. And then once we bind the ligand to the receptor, we get an enzymatic reaction. Protein kinases are one example of enzyme linked receptors where the enzyme itself phosphorylates a protein. It steals the phosphate from an ATP and adds that stolen phosphate to something else. This purple protein. Okay. All right. So that's what we're looking at with enzyme linked receptors. That is but one of a membrane receptor. Another type of membrane receptor is called a G protein coupled receptor. And all I want to say about these receptors is they basically are really good at setting off signaling cascades. They don't use ATP. Instead, they use another energetic molecule called GTP. So essentially, instead of adenosine triphosphate, it's guanosine or guanine triphosphate. I can't remember what it is, but it's G instead of A. But it's the same thing. You have a triphosphate, so high energy 3-phosphate group bond, and you use the energy in that third phosphate bond to set off a signaling cascade. So with G protein coupled receptors, what that means is that you have your receptor and it is in close proximity to a collection of proteins called, like collectively called the G protein. So this G protein is coupled to a molecule of GDP. But what happens is when you have your signaling molecule bound to that receptor, so you see that here, so your signaling molecule binds to the receptor, that causes the receptor to associate with that G protein. It couples with the G protein. And then what happens is you release the GDP, you actually phosphorylate it to create GTP, and that gives you enough energy basically to release a subunit or a component of that G protein. That would be the alpha subunit. And then the alpha subunit sets off a signaling cascade. So essentially, if we were to go back to this picture, what we would be seeing is a signaling molecule bind a receptor. This receptor would activate the G protein, and then this would be the alpha protein of the G protein complex, and it just sets off a signaling cascade. So that's all you need to know here. Enzyme linked receptors, you have the ligand bind the receptor, and a chemical reaction takes place right there. G protein coupled receptors, you have a ligand bind a receptor, the receptor then associates with the G protein dissociates, and then sets off a signaling cascade. So that's all you really need to know. It just adds a couple more steps, which again, like, why is this so complicated? The more steps, the more amplification of that signal, which GPCRs are like notorious for really good signal amplification. They are going to be one of the most common types of receptors in cell membranes. And they are also just give you a lot of steps for intervening in this process. So if something sets off this cascade and you're like, wait, that was a mistake, you have the space to say that, right? Which is really important to cells. They want to be able to change their mind. Okay. So enzyme-linked receptors, G protein coupled receptors. Oh, this is a student sent me this actually from the Monday, Wednesday, Friday class, but I still wanted to share it with you. This is just another kind of diagram showing the GPCR process. So I just included it for reference. So the other type of membrane receptor you can have is called a ligand-gated ion channel. So this kind of tells you exactly what it does. So these are integral proteins. These are channel proteins. But importantly, these channel proteins are closed. So notice that our ions in this case are like outside of the cell. They can't get through their designated channel. And let's say we want to open that channel, but we want to just tell the cell when to let those ions in. And we do that, basically what happens is this channel is closed until a ligand or a signaling molecule binds it, and binding of the ligand opens that channel and then lets that ion in. So ligand-gated ion channels basically are just like closed doors that if you have the key to the lock, open. I guess you could say that the signaling molecule is sort of like a garage keypad where you put in like your garage code and then the garage opens, right? You can't enter the garage without opening the garage door, and you have to put the keypad, the key code in to open the garage door. Yeah, I guess you could like drive your car through the garage, but that is not recommended. So ligand-gated ion channels are just like when those channels are closed, but binding of a ligand can actually open those channels so that the ions can pass through. Okay, so again, those are our three different types of cell surface receptors. So meaning if they're in the cell surface, that means they're in the plasma membrane. So enzyme-linked receptors mean that when a ligand binds a receptor, that receptor essentially functions as an enzyme to perform a chemical reaction. We talk about protein kinases, a really important type of enzyme-linked receptor that basically, like when they receive a signal, phosphorylate a protein. G protein coupled receptors are just like when when a ligand binds a receptor, it associates with another membrane protein and sets off a signaling cascade. Ligand-gated ion channels are ion channels that are closed, but when a ligand binds them, they open up to let whatever inside the cell. Now, we also have, so like with these cell surface receptors, basically all of these are interpreting signals that are outside of the cell and can't pass directly into the cell. Because remember that the cell membrane is selectively permeable. So not everything can pass directly through the cell membrane. The vast majority of ligands cannot pass through the membrane. And so they have to like signal through cell surface receptors. Some signals, especially hydrophobic or non-polar signals, for example, like steroids like estrogen and testosterone, because of the fact that they're nonpolar, they can pass directly through the membrane. And so these don't necessarily have to bind a cell surface receptor. They can actually bind receptors that are in the cytosol. So this is just showing, this is showing estrogen, which does have an intracellular receptor. And again, intracellular receptors are only used for molecules that can pass through the cell membrane. And again, this is because of their chemical properties that make them, the membrane, permeable to them, which is again is primarily nonpolar, small nonpolar molecules. So estrogen can pass directly through the cell membrane. Not only that, it can actually pass through the nuclear membrane and into the nucleus. And that's actually where their receptor is located. Their receptor is actually located really close to DNA, because once estrogen binds its receptor, that receptor actually is called a DNA binding protein. So once estrogen binds its receptor, it binds DNA and causes some sort of genetic change. All right, so those are your intracellular receptors. Okay, so this is where I get a little excited because, like I said, I hated cell signaling when I was in undergrad, but I learned to love it. One of the reasons I learned to love it is because cell communication is essentially how your body communicates, and how your body, or like how your body like gets messages and how your body responds to those messages. So as you all know by this point that I really love disease, and it turns out that disease really is just how your body reacts, or how your immune system reacts to something bad. So we talked about in the lectures from last week the plant communication, and we talked a little bit about plant response to attack. So I thought that since in our Muddiest Points we got to talk about the fact that we have many receptors in a cell, some of which are cell surface receptors, and some of those are intracellular receptors, I wanted to highlight some examples of those receptors, why location is so important. And I thought, what better way to do that than talking a little bit about how your cells, how your immune cells, actually all of the cells in your body, how they detect invaders, pathogens. So kind of like how plants detect bacteria or other pathogens. So in our lecture on plant communication, you learned about elicitors. And if you remember, elicitors were just like components of bacteria or viruses that were basically like interpreted by the cell and elicited a cellular response. So in the lecture, we talked about the production of nitric oxide and hydrogen peroxide, or even necrotic spots, which would be cell death. Your cells have very similar ability to detect elicitors. So this is a huge slide. Let's go through it. What you're looking at here is in gray is the cell plasma membrane. This would be I know it looks like the nucleus, it's not a nucleus, it's an endosome. So why this matters is because remember that your immune cells do have the capability of performing phagocytosis. So I emphasize that because an endosome would result from a cell undergoing phagocytosis. However, some of these are also located like in the cytoplasm as well. So in that plant lecture, we talked about the fact that like plant pathogens or disease-causing organisms are like their components are called elicitors. But when we talk about human immunity, we typically talk about what are called pathogen-associated molecular patterns, so or PAMPs. So the reason we say this is because we've kind of talked about like little bits about how your body, like there are, and probably you know, there are good and bad bacteria. So for example, E. coli that causes severe diarrhea or bloody stool is like bad E. coli. However, there is E. coli that is good E. coli and helps you produce really important vitamins in your GI tract. So how does your body distinguish between good and bad E. coli? And the answer is via pathogen-associated molecular patterns, or PAMPs. So for example, the bad bacteria have certain molecular patterns or really like minute characteristics that essentially make them bad. So your body is able to like recognize those small patterns as bad and sort of like not recognize patterns that are associated with good bacteria. There are several of these. I'm not going to go through them, but I do want to talk a little bit about some of them. So for example, there are certain lipopeptides that are associated with gram-positive disease-causing bacteria, which would be like staph infections. So like bad staph. LPS or lipopolysaccharides are associated with bad bacteria like E. coli or salmonella or cholera. We talked about flagella. So being a motility structure. So aside from sperm, which have a very distinct flagella, the majority of your cells, like all of them except for sperm, do not have flagella. And it turns out that actually a lot of the disease-causing organisms or disease-causing bacteria have flagella. And so you actually have receptors that detect flagella. So basically, if your cell sees a flagella that's not a sperm flagella, it's going to be like, this is bad news bears, right? So you have cell surface receptors that detect flagella. You have cell surface receptors that detect things like E. coli, cell surface receptors that detect like staph or strep. And what happens is those receptors detect it and set off a signaling cascade. You'll notice some of these, NF-kappa B, JNK, and P38, those are actually transcription factors that initiate inflammation or immunity. So those are cell surface receptors. Again, you also have several receptors that are intracellular. Some of them are associated with an endosome or a phagosome, same thing. And some of them hang out in the cytoplasm. But notably, what these detect is, you'll notice like, this is a little confusing, CpG-rich DNA, which is sort of like structurally more associated with viruses and bacteria. We won't go that much into depth. But I do want to mention that you have cellular receptors, intracellular receptors, that detect viral RNA. So they're able to tell the difference between your RNA and viral RNA. So like if, let's say like SARS-CoV-2 or COVID-19 virus gets inside of your cells are like, this is not human RNA. This is not supposed to be there. And it starts an immune response. Double-stranded RNA looks really weird. Your body does not have any double-stranded RNA. So if your cells see double-stranded RNA, they're like, this is wrong. And then viral envelope glycoprotein. So this is really important because remember that the viral envelope is made out of phospholipid membrane, just like your cell membrane. So how are we supposed to distinguish a virus from your own plasma membrane? And the answer is they do have certain proteins associated with their membranes, namely the ligands that bind to cell surface receptors and get them inside in the first place. So I just want to mention this. This also goes through kind of some of them. These would be your cell surface immune receptors. These would be your intracellular receptors. This is one type of cell receptor that is involved in immunity. These are called toll-like receptors, or TLRs. But again, I just think these are amazing because why cell signaling is so important is without these receptors, your cells wouldn't be able to tell the difference between your cells and a bacterial cell. Without receptors, your cells wouldn't be able to tell the difference between like good E. coli and bad E. coli. And this is so important. So one of the questions I get a lot is like, okay, so, you know, bacteria are obviously like recognized by the body as bad because they don't look like human cells. But how come when you eat like a banana, which is not human, how come your body doesn't react to that? That's not human, so why is it not recognizing it as bad? And the answer is because you don't have cell receptors that recognize banana proteins as bad. Some people do have cell surface receptors that detect certain things as bad. So these people would have maybe like food sensitivities in some case. That's just one mechanism of food insensitivity, but there's a lot. But yeah, so like cell receptors, I know it's complicated, but they're so vital because essentially it allows your cell to communicate messages, one of which is this is supposed to be here versus this is not supposed to be here, this is bad bacteria. Okay. So again, signal transduction is amazing. It's so important. And if you have a pretty decent formative understanding of it, ooh, you're gonna be good for like all of biology from here on out. So good job. All right. As usual, I just included some resources as well. We do have a couple of more minutes. That's all I have prepared for today because I know I was going to talk about soap, but we talked about that last time. I don't know why I didn't think about that. So I'm done. But I will certainly hang out for longer. If you have questions, I just opened the chat, or you can unmute yourself. But yeah, hang out if you want to, or if you want to go and get on with your day, you're welcome to do that too. It was so good to see you all.
Thank you.
Thank you. See you soon. Thank you. Thank you.
Have an awesome day, Professor.
You too. See you soon.
Thanks, Dad.
Good luck on your math exam. You're going to do amazing. I'm so glad the drink water down.
Dr. C.
Yeah, I'm so glad. Good.
I mean, I hope I do amazing.
You will. I'll be thinking all my positive vibes.
Because my math class isn't a traditional math class. It's like, because I'm in elementary education, so we're like learning the techniques of how to teach the math. So like I have to memorize all of that.
I need to take that class. Sounds like something I need to take.
It's actually really fun.
Good. That's wonderful.
Plus,
well, then you're going to do amazing.
My professor's kind of cuckoo, so he's like funny.
Oh, that's good. That makes sense. I personally love cuckoo professors.
Me too.
Well, good luck. I know it'll be.
Yeah. Bye, Dr. C.
Talk to you soon.
Talk to you
Dr.
soon.
C.
Yes.
Okay. Hi. So I'm just like trying to relate this like conceptually back to like the human body. So does every type of cell have every type of signaling, or is it like specific cells have specific signaling?
That is such a good question. And the answer is like cell signaling and what messages certain cells can get is actually what distinguishes cells. So for example, like in the human immune system, I don't know how much you know about the immune system, but do you know about like the fact that you have like two types of T cells, like CD4 T cells and CD8 T cell? It's okay if you don't, that's totally fine. So you have a type of immune cell called T cells, and these cells can do one of two things. Some of these T cells can basically tell the rest of the body, like, hey, there's something bad here, like prepare for war. And the other type of T cell, if it gets activated, it like tells cells like for example, if they're infected with a virus, this cell will like initiate killing that cell so that because without the cell, the virus can't go on. So the cells that are like basically like the generals of the army versus the cells that like kill cells are two different types of T cell, and they're characterized based on what type of cell surface receptors they have. So like their particular cell surface receptors are called, a lot of cell receptors are called like CDs. I can't remember, it's like cellular determinant or something. But CD4 cells, the cells with the CD4 receptor, are the generals of the army, and the cells with the CD8 receptor are the cells that like kill virally infected cells. So we actually like distinguish them by what receptors they have. That's a really good question.
Okay, perfect. Also,
Yeah.
do you watch Bob's Burgers?
I love Bob's Burgers. Yes.
Because you say like, all right, a lot. And I'm like, I see you.
I get it from that. And also I get it from that TikTok where the girl's like, all right, take it easy.
It is the, yes. Oh my God. Yeah.
But yeah. Yes, a lot of Bob's Burgers. Yes.
Okay. Well, have a good day. Thank you.
You too. See you soon. Excellent question. Any other questions? Everybody feeling good? All right.